Men and women are different, but they're not that different. But genetically now, this is hard to explain because women chromosomally typically have two X chromosomes, each X chromosome possessing about 1,200 genes, while each male typically has an X chromosome with 1,200 genes and a much smaller Y chromosome with only about 70 genes, which means that if genes were equally expressed in cells, then females would have double the dosage of the products of these genes. So that would be well over 1,000 genes, and females would have twice as many proteins and RNAs produced by these genes. This would lead to incredible biological differences between males and females. If genes were simply expressed in the same levels in each gender, then there would be enormous genetic differences due to the levels of proteins, enzymes, hormones, receptors, regulatory RNAs, etc. This difference in gene dosage is avoided because females effectively inactivate one of the two X chromosomes that they inherited from their parents in each of their body cells. This is done at random. So in half of a woman's cells on average, the X chromosome she received from her mother is inactivated, while in the other half that she received from her father is inactivated. There are genes encoding regulatory RNAs which initiate this process during embryonic development, and it occurs at random so that the X chromosome, which is inactivated, is condensed into what is known as heterochromatin, tightly bound DNA where genes will not be expressed. Although there are a number of tests which can probe for the presence of this tightly bound inactivated X chromosome, visually it constitutes what is known as a Barr body, where, for example, if one were looking at white blood cells in a blood sample from a woman, one could see these dense drumstick-shaped sections of DNA which one would not observe in males. These are known as Barr bodies and once again would indicate the presence of an inactivated X chromosome. One could also observe Barr bodies in other body cells as a dense region of DNA, smaller than the nucleolus, which is typically along the periphery of the nucleus. These Barr bodies might be of use in helping to identify the chromosomal gender of a child born with ambiguous genitalia, in identifying males who are XXY, which, although having a Y chromosome and being male, would also then have a Barr body because they have two X chromosomes, one of which would be inactivated, or an XXX female, where there would be two Barr bodies per nucleus.